Today we're learning more about mutations and the different types of mutations and the types of effects that mutations can have on the final product. So a couple questions here to consider. Why does it matter if mutated DNA produces a protein with a different amino acid sequence? So what effect would that change have on that final product? And even what effect might that have on the organism? Another question to consider is, do all mutations actually result in altered amino acid sequences? Throughout the lesson today, hopefully you'll be able to answer those a little bit more clearly. I want to go back to thinking about just amino acids in general. So there are 20 different amino acids. You may remember that from a couple units ago. Now, all amino acids have the same basic structure here. So you'll see that they all have these little pieces. The part that's different is those shaded pieces, and we call those R groups. So those are the variable groups in different amino acids. So notice that those variable groups change the characteristics of the amino acids. So some are nonpolar, and they repel water like oil. Some are polar and love water and are sort of attracted to one another. And others are electrically charged, making them either acidic or basic. So those different characteristics mean that the way that a protein actually folds into its three-dimensional shape is going to be affected by those R groups. They're going to fold in different ways. If we have a whole bunch of non-polar amino acids, they're not going to be mixing with polar amino acids. They're also not going to be mixing with electrically charged amino acids. So it changes the shape of the protein that's made. So in the last couple classes, you were looking at hemoglobin. So if we look over here on the left, this on the left side of this diagram is showing typical hemoglobin without a mutation and the shape that it's making. Over here, this is showing the mutated hemoglobin, and it changes the shape of that protein, which means the behavior of that protein is different, especially under low oxygen environments. So the difference was just a single base in the DNA. So that only changed one codon in the mRNA, and it changed one protein or one amino acid in the protein sequence. So the original hemoglobin had glutamic acid in there as one of the amino acids. In the mutated hemoglobin, it had valine instead. And so notice that we're switching one that's electrically charged and acidic to an amino acid that's nonpolar and repels water. So it acts very differently, and it causes that protein to change shape. Now, if the change had been from like glutamic acid to another acid, it may not have had as big of a difference or as big of an effect on that protein. So today you're going to learn about different types of mutations, and you'll see that the type of mutation can have a really big impact on the type of effect that the mutation has on the final product, on that protein. So good luck today. Let me know if you have any questions.